Hi, everyone. Arsalan Arif here with Endpoints News, and thanks for joining us today. Our topic is using external data to accelerate randomized controlled trials without introducing bias. This webinar is sponsored by Unlearn. Unlearn is the only company creating twintelligent RCTs, which combine AI, digital twins, and novel statistical methods to enable smaller, more efficient trials. Twintelligent RCTs incorporate prognostic information from digital twins into randomized controlled trials to enable smaller control groups and generate evidence suitable for supporting regulatory decisions. Unlearn's technology has been published in conference abstracts and scientific journals, including Scientific Reports, Nature. Unlearn is partnering with the world's leading pharma companies and continues to have discussions with regulators, including the FDA and EMA. Joining us today on the panel, we have Charles Fisher, the founder and CEO of Unlearn AI. Charles is a scientist with interests at the intersection of physics, machine learning, and computational biology. Previously, Charles worked as a machine learning engineer at LeapMotion and a computational biologist at Pfizer. He was a Philippe Meyer Fellow in Theoretical Physics at ENS in Paris, France, and a postdoctoral scientist in biophysics at Boston University. Charles holds a PhD in biophysics from Harvard University and a BS in biophysics from the University of Michigan. Also joining us today is Dave Miller, the head of biostatistics at Unlearn AI. Dave is a biostatistician with expertise in prognostic models, clinical trials, observational studies, and diagnostic devices. Most recently, Dave was head of biostatistics and epidemiology for Verily Life Sciences. Other past roles include lead program biostatistician at Genomic Health and senior director of medical affairs, statistical analysis at ICON Clinical Research. Dave received his bachelor's and master's in statistics from Carnegie Mellon. He has co-authored over 100 peer-reviewed papers in medical journals. If you have any questions during today's webinar, our panelists have reserved some time for questions. Hit the Q&A button at the bottom of your screen as soon as you think of the question. This webinar will be available on demand tomorrow to rewatch or to share it with your colleagues. And now I'm going to pass it off to Charles to get us started. Charles?
Thank you very much, Arslan. We're excited to be back here working with Endpoints on another webinar, and certainly encourage people to go back through existing webinars that we've presented with Endpoints over the past year. So we're going to tell you today how we can use external data to accelerate randomized controlled trials in a way that doesn't introduce bias. Now, when we're talking with clinical ops or medical directors from pharmaceutical companies, we hear the same complaints. We hear them say, starting and managing so many clinical sites, it just adds a tremendous amount of time and cost to our clinical trials. And we're competing against other trials for our patients. And as a result, recruitment is just too slow and our timelines are delayed. Now, every pharmaceutical company is dealing with these problems. And as a result, we hear a different set of issues coming from biostatistics groups, data science groups. They say, we are exploring how we can use external controls to solve these problems. But we're also struggling with the regulatory implications. What if the FDA and EMA won't accept the evidence from our study? So what exactly is this external control that I'm talking about here? So pharma companies have been exploring these techniques for many years in various forms: external control arms, synthetic control arms, historical control arms. So in a typical RCT, you randomly assign some patients to receive the active therapy, and you randomly assign other patients to a control group. In a trial with an external control arm, you delete the control group. Every single one of your patients is assigned to receive the active therapy. And then you leverage additional information about patients not receiving that therapy that can be taken from different sources: previous trials, real-world sources, registries, etc. And there's, again, lots of different names for this: external control arms and synthetic control arm, historical control arm, hybrid control arm. There are subtle differences between these different techniques, but for the context of this webinar, we are all just going to use one term, typically synthetic control arm. So the problem is that although these methods, they make your trial a lot smaller, they require unverifiable assumptions that there is a lack of alternative explanations between your group of patients receiving the active treatment and the group of patients in the external control. So the FDA says that this inability to control bias often makes the design unsuitable. Now, the problem is, you can measure lots of different characteristics, patients' age and weight and other types of things, and you can correct for that. But you can't correct statistically for the things that you haven't measured. And we need to be able to guarantee that we can have unbiased estimates of treatment effects, even in the presence of things that might be important that we can't or haven't measured. And the only tool that we know of that can do that is randomization. So we've developed a new way of running clinical trials that uses external data in a way that allows us to design more efficient trials, but maintains all of the benefits of randomization. We can think about placing clinical trials on this kind of 2x2 matrix. On the left are trials that are larger, require more subjects, and are hence slower. And on the right are trials that are smaller, require fewer subjects, and are faster. At the top would be trials that are very reliable, generate reliable evidence. On the bottom, trials that generate less reliable evidence. In the traditional randomized controlled trials over here in the top left, it generates very reliable evidence. That's why it's considered the gold standard approach, but it requires a large number of subjects to do so. So they take a long time and cost a lot of money. Down here in the bottom right are trials with a synthetic control group. You deleted the control arm, so it's much, much smaller, have fewer subjects, you can run it much faster. But at the same time, because there can be alternative explanations for the observed differences between your groups, you can't actually rely on the evidence from these trials without making some strong assumptions. So we're going to tell you today about something new that we call a twintelligent RCT. And this is a new kind of randomized controlled trial that is much smaller, but generates evidence with the same level of reliability as a traditional randomized controlled trial. So what exactly is a twintelligent RCT? It's a combination of 3 different technologies: artificial intelligence, digital twins, and the oldest technology, randomization. So first, let's walk through briefly how this works. A digital twin is a computer simulation of an individual patient that allows us to ask, how would this patient's disease progress if they did not receive the active therapy? So what would happen if they were randomized to a control group? And we can create one of these digital twins for every single patient when they enroll into a study, and then incorporate that information into the analysis at the end of the study in order to obtain powerful, unbiased estimates for treatment effects. Let me give you an example of what a digital twin looks like. This is a digital twin that we have created for a patient with Alzheimer's disease. When this patient enrolled, in a hypothetical patient enrolled into the study, we measured all of their different characteristics at baseline, as you normally would, input that into a pre-trained digital twin generator. And that predicts how every single one of these different measurements associated with that patient will change over the course of the trial. We can predict actually the whole distributions of those so that we can get means and estimates of the uncertainties and how all of those different characteristics would change. So a digital twin is a very rich, complete prediction of a patient's prognosis. And we create these digital twin generators by learning from historical patient data. So we take data from many different control arms of many different studies, and in some cases, observational studies and registries, and incorporate all of that information on a particular disease in order to build something that we can learn from. And then we apply novel proprietary deep learning methods to learn from that longitudinal patient level data, how to create a digital twin for a patient. Then we can apply this in the context of a clinical trial by taking that pre-trained and pre-specified digital twin generator and inputting the baseline data from a patient when that patient enrolls. And this outputs that patient's digital twin. And these digital twins can be seamlessly integrated into trial analyses using reliable statistical methods. In fact, statistical methods that biostatistics groups will be very familiar with. So this doesn't require significant changes to the way trials are run, but it can have a large impact. In Alzheimer's disease, we've demonstrated that we can reduce the size of control groups by 35%. That means fewer sites, less overhead, faster trials, and lower costs, while maintaining the same power as in a typical RCT. So just to summarize the introduction, we're going to tell you today in a bit more detail about twintelligent RCTs, a new design that enables high-powered studies with small control groups so that you can generate reliable evidence in a fraction of the time. And I'm going to turn it over now to Dave to walk you through some of the details on how we can do that.
Thanks so much, Charles. Both synthetic control arms and twintelligent RCTs use external data that may differ from the trial data. So why are twintelligent RCTs suitable for pivotal trials when synthetic control arms are not? Let's start by talking about what we want out of a study. We want a study to show that the investigational drug works. Simple enough. Or to be more precise, we want a study that shows that the investigational drug works in those cases where the investigational drug really does, in fact, work. Equally important, we want to be sure that the study doesn't accidentally show that the drug works when the drug does not in reality work. When this happens, we call it a type 1 error. I'm staying away from statistical terminology in this talk, but I do need you to understand what type 1 error is. It's a special type of mistake where we claim a drug works when it doesn't. And you can see why that kind of error would be of special interest to regulators. Now, there's also the possibility that our study shows the investigational drug doesn't work, and that if that's the truth, it actually doesn't work, that's okay. It makes us sad because we'd rather have a drug that works, but that's an appropriate decision. Lastly, we want to avoid situations where the study shows the drug doesn't work, even though it does work. Those studies are huge wasted opportunities, and it can happen when you didn't enroll enough people in your study. Both of the methods we're talking about today are effective at avoiding those missed opportunities. And Charles showed earlier how you can get a 35% reduction in the control arm and still avoid these missed opportunities with twintelligent designs. If you're interested in learning more about our methods, please see our previous webinars and white papers. The focus today is going to be on type 1 error, also known as false positives, because that's where bias from external data causes problems that make some methods unsuitable for pivotal studies. Type 1 error, basically the chance of claiming a bad drug actually works, is likely when there is bias. Bias here means systematically overestimating the true treatment effect. That's why the FDA says what it does and makes such a strong statement about the inability to control bias, that it's the major limitation of using externally controlled trials. This might sound like too big of a blanket statement, but let's look at a recent example from the pandemic. Consider these data published online in early 2020. The y-axis is the percentage of patients with positive COVID-19 tests. The x-axis is the days from the beginning of the study, and the red line represents patients who received hydroxychloroquine, and more of them got better every day. The black line represents controls, and they had very little change over the 6 days of the study. In summary, the hydroxychloroquine patients rapidly got better, while the controls did not. For those of you who like p-values, look at how low some of these are. This is amazing. And there's still an urgent need today for an effective COVID-19 treatment. Why isn't everyone using this amazing treatment, hydroxychloroquine? Well, they got the answer wrong. This wasn't a randomized study. There were non-random differences between the treated patients and the controls that created bias. For one thing, a lot of the controls came from a completely different hospital in another part of the country. It's easy to think of alternative explanations for why the outcomes would be different for those patients, reasons unrelated to hydroxychloroquine. And that's not all. Some of the controls were the people at the same hospital as the treated patients who said they didn't want to participate in the study. That creates a whole different set of alternative explanations for why the groups have different outcomes that, again, has nothing to do with the actual treatment. Bias and alternative explanations all add up to inflated type 1 error. What does this tell us about using external controls in general? Let's look at a simple example of people we could enroll in a study and see if we can see where the bias creeps in, and also see if we can control the bias. Remember, the FDA guidance doesn't just talk about bias, it talks about the inability to control bias. Each dot here represents a patient who could be enrolled in our study. That is, they've already been confirmed to have the disease we want to treat, and they meet our planned inclusion-exclusion criteria for the study. Now, even within the same disease, there will be some people in better health than others, and that's what this x-axis here represents. And pretty much every disease has some kind of health score that we can use to measure who's in better or worse health at the beginning of the study. In Alzheimer's disease, we look at scores based on memory and cognition. In irritable bowel disease, we assess abdominal pain. Some disease severity measures are more objective than others, but there's always something that we measure. There are also factors that could affect outcomes, represented by the y-axis, outcomes and patient selection, that we don't measure. A lot of it we don't measure because we just don't know about it. Even if we wanted to measure it, we couldn't. Some people have better doctors than others. And of course, it's not just doctors. They may have better overall care teams and may have better social support networks at home. They may have better access to quality care. They might also have some rare, as yet undiscovered biomarker that makes their disease progress faster or slower. In summary, the patients will differ in some ways that we can see and measure, and also in some ways that we can't see and can't measure and might be completely unknowable. So we couldn't even measure it if we really had our mind set on measuring it. So if we think about the outcomes for these patients, the ones on the top right are likely to have the best outcomes, with or without treatment. And the ones on the bottom left are likely to have the worst outcomes, with or without treatment. Keeping all of this in mind, let's suppose 6 of these patients are enrolled in a trial, and that's represented by these 6 green dots here. And let's randomize them. The solid dots represent the treated patients, and the unfilled circles represent the patients randomized to the control arm. Because the patients are selected at random, the control arm patient's health score is about the same on average as the treated patients. And the control arm patient doctors are equally good as the treated patients. And remember, I'm using better doctors here just as a proxy for all of the things we don't measure and can't see and might be completely unknowable. Because we randomized, there's no bias with respect to the stuff we know on the x-axis, and almost like magic, there's no bias with respect to the stuff we don't know about, represented by the y-axis. But what if we selected our treated population differently? Let's come back to our blue dots and start our selection process over again. Suppose only treated patients are included in our trial, represented by the 3 dots that just became green. And the untreated patients are selected separately, represented by the unfilled circles. If you select them differently, they're going to be different. Now, when I see different outcomes for the treated patients and the controls, even if those differences are statistically significantly different, I don't know if the difference is because the treatment works or if it's because of bias. But let's get a little nuance just for a minute in talking about bias. After all, the FDA guidance doesn't just say bias is bad and leave it at that. The guidance says the inability to control bias. Health scores are something we can measure in various ways for both the treated patients and the controls. So we know how different they are and we can make adjustments. I can argue a long time about the quality of those adjustments, but that doesn't matter very much. If you use the best available techniques, the adjustments can be pretty good. In other words, those ugly words, the inability to control bias, aren't necessarily an Achilles heel for known measured biases. Phew, maybe bias isn't so scary after all. But wait a second. What about the stuff we don't measure? There will be differences there too. And what's worse, we will have absolutely no idea how big or small those differences are. Remember the COVID hydroxychloroquine researchers? They measured symptoms and demographics, but there were probably other differences, other alternative explanations, which we'll never ever know about between the controls at the other hospital and the treated patients. There were also probably systematic and difficult to measure differences between those who opted out of treatment and those who wanted to try the new treatment. I'm saying probably a lot. It's not just because statisticians like to say probably. We just don't know. Maybe there are differences, maybe there aren't, but we don't know, so we can't say. This is the real reason for that scary, ugly phrase, the inability to control bias. Bias isn't terrible and scary all by itself. It's the inability to control bias that makes it scary and leads to unsuitable designs. Remember, unsuitable is the FDA's language. Now, let's come back to the randomized study. Remember, randomization is our friend. Randomization addresses both the biases we see and the biases we don't see. In this example, we've avoided bias through random assignments of treatment and controls, but it's a very small study, so I'm going to have trouble finding differences. If only there were a way to use AI and external data to make my study more powerful without adding bias. There is. That's what a twintelligent RCT is about. Twintelligent RCTs are built on two foundational technologies. The first addresses the question, how will this patient's disease progress without a new treatment? Every enrolled randomized patient in the study, represented by the woman in the color photo, gets a digital twin, represented by the shaded blue photo. The digital twins are created from external data, but twintelligent RCTs apply that model to the randomized patients. The second technology addresses the question, how can we use this rich data from digital twins to reduce the sample size for RCTs? I promised to go light on the statistical terminology, so I'm going to skip right over all these Greek letters that are essential to our patented methods. Suffice to say, they guide you to the smallest sample size. What's even more important is they allow us to use the added information from the digital twins to reduce the sample size without adding bias. With these two powerful technologies in mind, let's come back now to our example randomized patients. We use the AI that was developed from the external data, and instead of creating new potentially biased control arms, we apply the model to the randomized patients to get more valuable information about each patient. The added information is generated from the randomized patient's data before any follow-up occurs. So the twintelligent RCT inherits all of the essential properties of randomized trials for avoiding bias. I've thrown a lot of dots at you to try to make the case about how one method, the twintelligent RCT, can control bias, and another method, synthetic control arms, cannot control bias. But my examples so far have been on a small scale. What about a more realistic example? What if we think about a study of hundreds of patients with a much larger set of external data? With a more realistic example, what happens when important data associated with alternative explanations are unmeasured? We can simulate this. Create two sets of patients, trial patients and external data. Use the external data to make digital twins or synthetic controls. Run the study both ways and compute the key statistics. The type 1 error is the percentage of studies that indicate that the drug worked when we've simulated no treatment effect. Then we're going to repeat this experiment with bias coming from different sources, sometimes with most of the bias coming from measured variables and sometimes coming from unmeasured variables.
Dave, I apologize to interrupt. I think that the slides are not currently advancing.
Oh dear. Okay, thank you.
Yes, I maybe try to unshare and reshare.
Yep, thank you.
To get to your point.
Okay. Okay, thank you for that. Is it showing that we can simulate this with four points?
Yes, it is now.
Okay. Thank you very much. I'll just briefly say again, so we can simulate this, creating two sets of patients, trial patients and external data, use the external data to make digital twins or synthetic controls, run the study both ways and compute the key statistics. And then the type 1 error, remember that's the important statistical terminology here, is the percentage of studies that indicate that the drug worked when we've simulated no treatment effect. And I'm hoping it advanced this time. The y-axis of this graph is the type 1 error. Remember, that's the one statistical term that's really essential to understand the big difference between the two methods we're talking about today. The percentage on the y-axis are the chance of being wrong. For a single study, the FDA and most scientists are willing to accept that in a good experiment, that chance could be as high as 5%. And that's represented by the dotted line here. If the bias comes almost exclusively from measured data, our simulation suggests that you really can control that bias over on the left-hand side, and you won't be wrong too often. On the other hand, if the bias comes from unmeasured alternative explanations, then the error rate is literally off the chart. You're wrong more often than you're right. Of course, you're more likely to be in the middle, but what we see when the bias is an equal mix of measured and unmeasured variables is that the probability of being wrong is about 25%, which is far too high for regulators or other scientists. To intelligent RCTs get the benefit of randomization to manage measured and unmeasured biases equally well. If you look at the blue line, which is a simulation for the to intelligent RCT, the estimated type 1 error rate from our simulation is very close to 5% on the left, very close to 5% on the right, and very close to 5% in the middle. The to intelligent RCT controls bias for exactly the same reason that RCTs generally control bias. There's no alternative explanation for the treatment differences. Now, we do need to acknowledge that to intelligent RCTs are more effective, not just unbiased, but also effective when the AI is built with good measured data. Charles showed us this figure earlier. The traditional RCT is reliable, but has to be very large. The trial with an external control arm or a synthetic control arm can be smaller, but it's not reliable. Our simulation showed that the intelligent RCT is always reliable, regardless of the sources of bias. But the amount we can reduce the sample size depends on the quality of the digital twins. If the digital twins are a little bit less predictive, we can still reduce the sample size, but we can reduce it less. With exceptional digital twins, we can reduce the sample size even more, but always avoiding bias and always being reliable. This is an example of how higher quality digital twins can produce bigger benefits. In a recent collaboration with a large pharma company, we found that the digital twin quality was so good it would translate to a 35% reduction in the control arm in a future study. And this is for a study using our Alzheimer's disease model and using ADAS-Cog as the primary endpoint. The twintelligent RCT is the only method that can reduce the size of control arms without introducing bias. This is because the external data is incorporated into the study design and analysis, leveraging everything that is good and right about randomization in a way that is consistent with existing regulatory guidance. Twintelligent RCTs keep randomization as the centerpiece of the study design. This is because randomization addresses both known and unknown, that is measured or unmeasured, variables that affect outcome. There's a common misconception that you can address unmeasured variables by just doing a better job with data collection, but there are two big problems with that approach. One is that you usually have no control over the data collection for the external data, but the second is that most unimportant measured variables are unmeasured because nobody even knows what they are or how to measure them. That inability to eliminate systemic differences between treatment groups is the big unresolvable problem for studies with no concurrent randomized control. And that's why traditional concurrent randomized controls are a central feature of the Twintelligent RCT. The goal is to minimize the number of patients who need to be randomized to control rather than removing the central element of randomization from the design. That is what allows us to use the rich troves of external data that are increasingly available to researchers. To summarize, we've solved the problem of how to leverage external data to get to smaller, faster studies without creating bias. The struggle is over. Twintelligent RCTs deliver smaller control groups than traditional RCTs. Our patented methods and publications show that digital twins provide the maximum benefit achievable without adding bias. The combination of adding speed without adding bias is what makes the Twintelligent RCT uniquely suitable for pivotal studies. Digital twins are seamlessly integrated into clinical trials using established statistical methods that have been developed to align with regulatory guidances. For example, the recent guidance on covariate adjustment recommends adjusting for whatever is most strongly associated with the outcome of interest. Because digital twins were specifically designed to predict the full range of outcomes for each therapeutic area, they're ideal for this application. This is the optimal solution for how AI can help to reduce the size of control arm without adding bias. Now, what would the world look like if everyone used Twintelligent RCTs? Medical research could be dramatically accelerated. Fewer total patients would be needed for every study, and sites and participants would be more excited to enroll in a study where there was a much smaller chance of ending up in the control arm. All of this would lead to new, better treatments reaching the market and reaching patients sooner. Please reach out to us to discuss how we can accelerate your clinical development programs with Twintelligent RCTs. We're excited to work with you to get good drugs to the market faster, to get effective treatments to more patients sooner. Ask us also about our recent publications, one of which was just recently accepted and will be available online soon. And thank you very much for your time. I'm gonna hand it back now.
Thank you, Dave. All right, well, let's move to the Q&A session. Lots of good questions here from the audience, and let me encourage everyone in the audience to keep getting those in here for Charles and Dave. Let me get started off here for a question for Charles. Here, question from the audience. Has this approach been used in oncology? If so, what indications?
That's a great question. So the approach that we've developed can be applied to any disease area. But as a small startup company, we have to focus some of our efforts on initial things. So our initial efforts have been focused on neurosciences and immunology. So working in areas like Alzheimer's disease, Parkinson's, ALS, Huntington's, and now starting to do some new work in immunology, things like lupus and rheumatoid arthritis. So those have been the areas that we have worked on in the past and are currently working on, but we are definitely open to discussions and partnerships where we apply these methods to new disease areas. So if you have a particular disease area, whether that's in oncology or rare disease or any other kind of indication, and you want to know how this could work for you, please reach out to us. We'd be happy to discuss how we can make that work.
Dave, let's move on. Here's a terminology question from the audience. The question is this: could you compare or contrast propensity score matching and synthetic control? Are they one and the same?
So they're not one and the same. Propensity score matching is a way to get really an ideal synthetic control accounting for all of the measured variables. And in fact, in the simulation, I skipped over an awful lot of details, but the simulation did use propensity score matching for the creation of the synthetic controls. in that particular example. But there are various ways that one could come up with a synthetic control arm.
Okay. A question from the audience, Charles. Okay, so CNS trials are notorious for the placebo effect, especially in depression and autism. Can you please comment on how this model minimizes the impact of the placebo effect?
So when we think about the behavior of control groups, there are actually quite a few different things that go into that, right? So there's the placebo effect, which is really the power of suggestion, right? So patients may get better or worse because they think that they are receiving some new kind of experimental treatment. But there's also just natural history, right? So some patients just happen to get better or get worse, and that's just the natural course of the disease. And then because of the way patients are enrolled and screened into clinical trials, you often have a kind of regression to the mean effect. We're enrolling patients who have you have some cutoff for disease severity that allows you to get into the trial. And anytime you enroll people based on a cutoff like that, you're going to tend to see some regression to the mean. And so you have all of these different things layered on top of each other, leading to the behavior of control groups. And so our aim is actually to model all three of those things in the digital twins. And the way that we do that is by using data to train these models that have been collected from the control groups of previously run clinical trials. So the patients, the training data that we're using come from patients who are subject to all three of those forces. They are in placebo groups so that they were subject to some sort of placebo effect potentially. They obviously were enrolled into these studies and screened in, so they have the normal kind of regression to the mean effect. And there's the natural course of the disease underlying all of it. So really, we try to construct training data sets that have all of those different effects so that they are learned by these digital twin generators and can contribute to reducing variability in the twin-intelligent RCTs.
Okay. Here's a follow-up question, though, on that. Doesn't the knowledge that the patients are very likely getting the active arm inherently affect the results of a study where synthetic control is used for over 50% of your placebo arm?
So there's a really interesting literature on this as to looking at how how randomization ratio contributes to the placebo effect. And in that particular case, where I've seen the studies that have looked at this, typically there is not significant changes in the placebo effect as you vary the randomization ratio over reasonable levels, right? I think that when you start getting into truly open label trials, right, what we're talking about with synthetic controls are often things that are truly open label studies, which are unblinded, in which every single patient receives the active treatment. And that is a situation where you run the risk of potentially having large placebo effects. That's one of the things that's very difficult to measure that contributes to these alternative explanations for what you observe. But twin-intelligent RCTs don't have those characteristics. They are double-blinded, randomized controlled trials, just with unequal randomization ratios. So they minimize a lot of those types of problems that you do typically see in open label studies.
Let me combine, as usual, when I have you on these webinars, Charles, there's so many of these questions. And we always get this question, but I'm going to combine it for you and give you the opportunity to answer this again. Has the FDA already approved drugs with the twin-intelligent RCT approach? And do you have any experience with this being accepted by the EMA? There's several FDA and EMA questions.
Of course, there always are. It's the big question. We're going to have, we hope, some really big news on this in the future. For today, what I would say is we have used this approach in phase 2 and phase 3 clinical trials. We are currently partnering with companies in some studies that will be kicking off next year in phase 2 and phase 3 clinical trials. So we are indeed using this with companies now. Have any of those trials resulted in an approval of a therapy? No, that has not happened. Of course, clinical trials take a long time. Our company's been around 4 years. That's from inception, that's like the whole length of a clinical trial, right? And we don't make the drug work or not work. That's all. Ultimately, that we just want to discover help discover whether the drug does or does not work as efficiently as possible. It's up to the pharmaceutical company and the biologist to be able to develop a drug that does work. In terms of the EMA, we are currently going through qualification procedures in Europe. And so we anticipate to have some news on that in the future.
Great. Best of luck with that. Dave, some statistical questions here for you, I think. So here's a broad question, but let me throw it out and see if you can field it. Can you provide more details about the digital twin generator and how it works?
Well, some of it is proprietary, but I think some of it also has been described in a variety of our publications. But I will say that the way that it works is to use the historical data to get a complete picture of a patient, to use all of the known associations, all of the discoverable associations between all of the data that we have from these longitudinal records, to be able to create the generator. And then when we've got a new study, we get the baseline data for the new study and we plug that into the generator. So on the biostatistics side of the organization, we're mostly doing that second half of the work of taking the baseline data and plugging it in so that the use of these models is actually very straightforward for biostatisticians. And a lot of the very complicated and hard work is done from the data science and machine learning side of our organization.
Okay. All right. Here's another stats question for you. I'm going to read the question as is. What causes the up to 20% bias in the presence of measured variables?
I think that's referring to the simulation that I showed, and it's where I referenced there being a 25% bias, basically, or maybe it's 20% if you do the 25 minus 5. That's for a situation where there's an equal proportion of the confounding that's coming from the measured variables and the unmeasured variables. So it's an equal mix of measured and unmeasured. As I mentioned, you can usually do quite a good job with propensity scores or other methods to adjust for the measured variables. It's the unmeasured variables, which are inherently unknowable. So you've got no way of knowing if you've got a 50-50 mix or not. It might be 25%, it might be 75%. You've got absolutely no way of knowing. So you have to address the situation where it could be 50-50. And I think 50-50 is actually pretty plausible for what you know and what you don't know that could create these confounding effects. But it's the unmeasured variables. It's what you don't see.
Okay. Here's a question on validating the accuracy of the AI model of disease progression. Okay, so how do you validate the accuracy of the AI model of disease progression in the necessary and perhaps itself somehow unusual subset of patients selected for the study in question? Are comparisons made to confirm accuracy using the enrolled placebo subjects?
Charles, do you want that one, or you want me to take that one?
You can go ahead, Dave.
Okay. So all of our models undergo rigorous out-of-sample validation. In other words, data that was not used to train the models is used to assess what the performance characteristics are of the models. And we try to do that as often as possible with an entirely new study that comes along. And a lot of the people that we partner with are interested in looking at a comparison versus an older study or some data that they have available to get things going, to get a sort of general sense of what the overall model quality is. In terms of the actual study it's used in prospectively, we do have to make a guess as to what the quality will be, but we feel that they are very, very good guesses because of the extent of the validation that we've done on a variety of different samples to understand the performance characteristics of the model.
Okay, wonderful.
Dave, I would just add to that. I think that it's important to understand that the validation of the model or the lack of validation of the model, neither one of those situations would affect the type 1 error rates of a twin-intelligent RCT. Neither one of those situations reflect the bias of the treatment effect in a twin-intelligent RCT. So what we have done is created a clinical trial design for which you can incorporate digital twins from a model and mathematically guarantee that in all circumstances, you end up with an unbiased estimate of the treatment effect. In this paper that Dave pointed to in the International Journal of Biostatistics proves that rigorously. And so when we talk about validating a model, this is actually for us a part of study design, because the quality of the model, that translates to the reduction in sample size that we can provide for our clients. So what we're basically doing is we're measuring the performance of the model so that we can do that sample size calculation for our clients. And the better the model is, the smaller the trial they have. But they always end up with an unbiased estimate of the treatment effect.
Right. Well, here's this question that just came in as you were speaking, Charles. Have you invented methodology to calculate the needed control sample size, or the already existing methods would still work for this new concept? Right. So you've just addressed that.
Yes.
Yeah, you just addressed that. So wonderful. Next one. So can this approach be applied to open label trials with active control?
So the short answer is yes. The expectation is that, well, first of all, you're going to always have control over your type 1 error. You're never going to be introducing bias. But then the question is, how much can you reduce the sample size? And it basically comes down to how well we're discriminating between the people who are likely to do better and the people who are likely to do worse. And even an active comparator, that discrimination is largely going to be the same people, that the same people would do better and the same people would do worse with an active comparator versus a strict placebo. And so therefore, the methods are both absolutely totally unbiased with an active comparator, but also still effective at reducing sample size.
And also, just to add there as well, from the modeling perspective and the creation of these digital twins, we do incorporate potential medications that patients can be on, as long as we have data from patients receiving those medications, right? So if it's a brand new thing, it's never been given to anybody before, we can't predict how people would respond to it. But if we have data sets of patients on that active comparator, then we can also incorporate that directly into the predictions of the digital twin.
Right. Question here. What's the conventional ratio of number of subjects in the experimental group in the control arm? 35% smaller control is the max question. What's the average percentage that can be improved?
So I always say, we ask this question all the time. And unfortunately, there isn't like a rule of thumb that you can translate from one disease area to another. It's an empirical question that depends on the performance of the model. So if a model is very, very good at predicting the progression of the disease, then you might be able to have really substantial reductions in the size of the control group. If the model is not very good at predicting the progression of the disease, then your reductions are smaller. And so we ultimately have to do a calculation, we have to do an experiment. I don't mean run like an experiment in which we actually like run a real, but like we have to run simulations in order to estimate what it's going to look like for a particular patient population. I mean, that's part of what we do during study design. And so when we partner with customers, we typically are actually constructing those partnerships in a way where we're sort of like a tiered structure, where the more that we can reduce the clinical trial, the more like that would cost. But if we can't reduce it very much, it doesn't cost anything.
So what's the minimum? I'm sure what's the minimum N needed to generate to build a model. And what's your recommendation if you don't have a large historical data set?
Yeah, another question that we get asked all the time, and another one that I answer exactly the same way as the previous one, which is that there is no rule of thumb. It's an empirical question. In order to figure it out, you have to partner with us, and we can do an analysis to see if it's worthwhile pursuing. And it may not be, but it may be. We just can't say necessarily. In the disease areas that we're currently working on, we typically have data sets that are, on the order of 1 to 10,000 patients. That's kind of what we're working with. But we are following each one of those patients for some period of time. That's matched to the sort of typical rate of change of patients in that disease, right? So it might be different for different, but we want to have longitudinal data from 1 to 10,000 patients. That's a pretty good sweet spot for us.
A question here from the audience comes in. Does your methodology use our real world data for control group baseline?
Our methodology could use a digital twin generator that has been trained on data collected from routine practice, what people call real world data. Or it could be, use historical controls or register, or a combination of all of those things. But it's ultimately what we're not doing is what a synthetic control is you take data and you jam it into your clinical trial. So you take data from some other place and you like shove it into your clinical trial and you pretend that it was randomized in. And that's not what we do, right? So what we are doing are actually running real randomized controlled trials and then incorporating this information through predictions about the patients.
Okay. So how is your approach similar or different from methods for controlling, confounding, including frameworks like propensity scores, potential outcomes, or directed
DAGs.
Yep.
Directed acyclic graphs.
Yep. Acyclic graphs. There you
go. So one of the key factors that's involved in all of those approaches. So those approaches actually really come out of the literature in econometrics and epidemiology in areas where people are running a lot of observational studies. And the main point is that how can you, if you have an observational study, how can you try to tease out treatment effects from that study? And so, they require you to make assumptions. If you're interested in, especially the DAG approach or the philosophy of this, I really recommend Judea Pearl's Book of Why for going through. It's a great popular book about causal inference. And his ladder of causation is all about that you need to make assumptions. You need to make assumptions in order to tease causal effects out of observational data. There's no way that you can get it from the data alone. But if you do randomized experiments, that's not true. So randomized experiments allow you to do causal inference in some of these ways that Dave explained without having to make a lot of assumptions. I don't know, Dave, do you have anything you'd want to add to that?
Yeah, I mean, the methods that, that you're talking about all work with the measured data. They can't do anything about the unmeasured data. Sometimes people like to make assumptions about how correlated the measured and unmeasured data are. I actually baked that into the simulation. In the simulation, one of the little hidden parameters is that I assumed that there was a fairly high correlation between the measured and unmeasured variables. But even with that high correlation, there's still a lot that you don't know that can't be explained, that's hidden from you. And really only randomization works to account for that.
Okay. We just have a couple minutes here left, so I want to ask several more questions from the audience. Here's one. Should we expect to have different models for different countries, knowing that most of the RCTs are from different countries?
We typically believe that it's best to build one model because you're going to have a lot of information from one country that will also tell you about how patients in another country may react. And may progress. But we do, in some cases, include geographic regions or countries as variables in these models. So we try to account for that, but within the framework of the digital twin generator model.
Okay. I just want to read a comment posing as a question over here. Interesting approach. We'll be in touch to determine feasibility. Great. I'd love to see it. Let me ask another question here coming in. Single arm approaches have been gaining ground in FDA approval. For example, the recent Prograph approval that uses RWE control arm data. Can you comment on how your approach will work in situations like this?
So our main goal is to be able to develop an approach that can be applied not just in special situations like label expansions or ultra rare indications, but how do you get to the big, large billion-dollar RCTs that take 10 years to run? And those approaches that are appropriate for these special situations, just they're not suitable for these larger trials. And so that's really the huge bottleneck. 50% of clinical trial spend is spent at phase 3, is spent on pivotal trials. And so we really want to be able to focus on that bottleneck and make sure that we can address it. Do we have methods that would also work maybe that are a bit more aggressive for ultra rare indications or special situations? Yes, we do. So if that's the situation you're in, we'll be happy to talk with you about how we can do that as well. But this particular point was on how we can take similar approaches, make them safe and to be used and suitable within these large pivotal clinical trials.
Okay. Another question just came in, Charles. I'm going to field this right at you. How do you plan to sell this to regulatory agencies? I'm sure that they would like to understand the method or code itself in action.
Sure. Yeah. No, our papers, we publish academic papers that describe the approaches that we use. And so, as we mentioned, we'll have a paper that's in press now. It'll be coming out soon. And we are going through currently qualification procedures with the EMA. So, we have had meetings in the past, multiple meetings with FDA. We've had a lot of meetings with EMA as part of our qualification procedures. We have academic peer-reviewed publications. And frankly, that's why we patent stuff. Right? We patent our methods because when methods are patented, you're able to discuss them. So, if people want to actually dig into the science, I mean, this is a webinar, so we're not like throwing up a bunch of equations and doing derivations on the blackboard. But if people do want to do that, we can do that. And please reach out to us to have more detailed discussions.
Great. Yeah, a couple more comments coming in. One, I like to connect with your group. Love to see it. So let me finish this up. Let me ask one last question here, because I think we just have time for one last question. Does your system compensate for anomalies in entry subject variables? Won't it amplify deviations in numbers in smaller studies, assuming that it uses starting variables from random subject of the main arm?
Dave, I'm going to send that one to you.
I guess it wasn't entirely clear to me from the question if the anomalies are like real, actual outliers because the patient is in fact an outlier, or if it's a question of messy data. In the process of generating the digital twins, we do occasionally run into a value that looks to us to be a deeply suspicious value that we think maybe warrants some additional monitoring. And, that has come up, and sometimes we've found some, issues with data cleaning that it's just a sort of little tiny side advantage of using these methods is it's sort of an extra step that might help you find some of those anomalies and correct for them. If they're real anomalies and the patient really is an outlier, then the model will predict some outlier behavior that is probably correct.
Okay. All right. Well, that's all the time that we have for today. Charles Fisher, Dave Miller, thank you so much. I enjoy speaking with you so much. And I want to thank the audience too for taking their time, for spending the time with us today. We appreciate Unlearn for sponsoring the discussion as well. And if you'd like to rewatch the webinar or share it with your colleagues, a link for on-demand viewing will be sent tomorrow. I'm Arslan Arif for Endpoints News, and thank you for joining us.